Close your eyes and imagine the smallest library in the world. It sits quietly inside every cell of your body. Right now, as you breathe slowly and settle into comfort, billions of these tiny libraries are working in perfect harmony. This library doesn't hold books made of paper. Instead, it holds the most important story ever written. Your story. Written in an alphabet that contains only 4 letters. These 4 letters have names that sound like a gentle chant. A. T, G, C. Adenine. Thymine. Guanine. Cytosine. Say them softly in your mind. Let their sounds roll like waves across your thoughts. For nearly 4 billion years, these 4 letters have been writing the story of life on Earth. They wrote the first simple bacteria that learned to eat sunlight. They wrote the ancient fish that grew legs and walked onto land. They wrote the first flowers that painted the world in color. And now, they write you. Imagine these letters as tiny dancers, spinning and twirling in pairs. A always dances with T, G always partners with C. They hold hands across the width of a spiral staircase so narrow that millions could fit across the period at the end of this sentence. This spiral staircase is your DNA. It twists gently, like smoke rising from a candle. Like the curve of a seashell. Like the path of a vine reaching toward sunlight. Each rung of this spiral ladder holds 1 pair of dancing letters. Your DNA contains over 3 billion of these rungs. 3 billion dance partners, spinning slowly in the darkness of your cells. If you could stretch out all the DNA from just one of your cells, it would reach 6 feet long. Yet it's packed so carefully, so perfectly, that it fits inside a space smaller than the head of a pin. Your body contains about 37 trillion cells. Each one holds this same 6-foot strand of DNA. If you could lay all your DNA end to end, it would stretch from Earth to the Sun and back again. 60 times. Think about that as you sink deeper into relaxation. Inside your quiet, resting body lies enough genetic material to span the solar system. You are vast in ways you never imagined. But these letters don't just sit quietly on their spiral staircase. They speak. They whisper instructions in a language older than mountains. Older than oceans. A language that your cells understand perfectly, even though you've never learned to speak it consciously. This language tells your heart cells how to beat in rhythm. It tells your lung cells how to capture oxygen from the air you breathe. It tells your brain cells how to dream, how to remember, how to love. The words in this ancient language are not long. Most are only 3 letters at a time. ATG might mean start here. TAA might mean stop here. Simple words for complex miracles. These 3-letter words spell out instructions for making proteins. Proteins are the workers of your body. Some proteins carry oxygen in your blood. Others fight off infections. Still others help you digest the food you ate for dinner. Your body makes over 20,000 different types of proteins. Each one is built by reading the genetic instructions written in your DNA. Like following a recipe, but instead of making bread or soup, your cells make the very substance of you. As you rest here, your cells are reading these recipes constantly. They're making new proteins to replace old ones. They're repairing tiny damages from the day. They're preparing for tomorrow's needs. This process never stops. Not when you're awake, not when you're asleep. Right now, as your breathing slows and your muscles relax, millions of cellular chefs are following genetic recipes with perfect precision. Some people call DNA the blueprint of life. But blueprints are flat and lifeless. Your DNA is more like a song. A 4-note song that plays billions of variations. Each variation creates something new. A fingernail. A freckle. The curve of your smile. Scientists have learned to read this genetic song. They can spell out the sequence of A's and T's and G's and C's that make up your genome. Your personal genetic library now fits on a computer hard drive. But reading the song and understanding its meaning are very different things. We're like children who have learned to sound out words but don't yet know what stories they tell. Your genetic story began long before you were born. Half of your letters came from your mother, twisted into her DNA like music in a jewelry box. Half came from your father, carrying melodies from his ancestors. When these two sets of genetic music combined, they created something entirely new. You. A song that had never been sung before and will never be sung again in exactly the same way. Yet you share most of your genetic alphabet with every human who has ever lived. You share large portions with mice and elephants and apple trees. The language of DNA connects all living things in one vast, ancient conversation. Your DNA remembers when your ancestors lived in trees. It remembers when they first used tools. It remembers migrations across continents and adaptations to new climates. This memory lives in the spacing between letters, in the rhythm of repeated patterns. But your DNA doesn't just remember the past. It dreams of the future. Each time your cells divide, they copy these four letters with extraordinary care. They're preparing the instructions that will build tomorrow's you. Sometimes, very rarely, a letter gets copied incorrectly. A becomes G, T becomes C. These tiny changes are called mutations. Most do nothing at all. But occasionally, a single letter change can alter the entire meaning of a genetic sentence. This is how evolution works. One letter at a time. One small change leading to another, across millions of years. The same four-letter alphabet that wrote the first life on Earth continues to write new chapters every day. Now humans have learned to edit this ancient text. We can change individual letters with tools more precise than the finest surgeon's blade. We can rewrite genetic sentences to fix errors that cause disease. This power to edit the alphabet of existence brings us to the edge of directing our own evolution. We stand at the threshold of becoming co-authors of the genetic story that has been writing itself for billions of years. As you drift toward sleep, feel the quiet hum of your cellular libraries. Feel the ancient dance of four letters spinning in perfect spirals. Know that you carry within you the wisdom of ages and the potential for futures not yet imagined. Your DNA whispers its secrets in the darkness, and somewhere in that whisper lies the key to rewriting tomorrow's world. Picture yourself walking into the most magnificent library ever built. The ceiling stretches beyond sight. Shelves upon shelves hold billions of volumes, each one containing the secrets of life itself. This library exists inside every single cell of your body. Right now, as you breathe slowly and let your muscles relax, 37 trillion of these libraries are quietly humming with activity. But this is not a library made of paper and ink. The books here are written in molecules. The shelves are made of proteins. The librarians are tiny machines that never rest, never sleep, never make mistakes. Your DNA sits at the heart of this cellular library like a master catalog. It contains every instruction needed to build and maintain you. But DNA is precious and fragile. It never leaves the safety of the cell's nucleus, protected behind multiple walls like a priceless manuscript. So how do these instructions reach the parts of your cell that need them? How does the recipe for making insulin get from the DNA in your pancreas to the protein-making factories in the cell's outer regions? This is where RNA enters the story. RNA is DNA's faithful messenger. Think of RNA as a careful librarian who makes copies of important pages and carries them wherever they are needed. RNA looks similar to DNA, but with small, important differences. Where DNA has the letter T, RNA uses U instead. Where DNA is a double spiral, RNA is usually a single strand. These differences make RNA more flexible, more willing to travel, more ready to work. When your cell needs to make a particular protein, it sends a signal to the DNA. The DNA responds by unwinding slightly, like opening a book to a specific page. Special enzymes read the genetic instructions and create an RNA copy. This process is called transcription. Imagine a medieval monk carefully copying a manuscript by candlelight. But instead of taking hours, your cellular scribes can copy a gene in minutes. Instead of using quill and ink, they use molecular brushes and chemical paints. The RNA copy carries the genetic message out of the nucleus and into the cell's main workspace. Here, remarkable machines called ribosomes wait like patient translators, ready to turn the RNA message into a living protein. Ribosomes are among the most ancient and essential machines in your body. They exist in every living thing on Earth, from the tiniest bacteria to the largest whales. They are older than eyes, older than brains, older than blood. Each ribosome reads the RNA message 3 letters at a time. UUU means add the amino acid phenylalanine here. GGG means add glycine. CAU means add histidine. The ribosome follows these instructions like a chef following a recipe, adding amino acids one by one. As the amino acids join together, they begin to fold into complex shapes. Some fold into spirals. Others form flat sheets. Still others twist into elaborate knots and loops. The shape determines what the protein can do. Some proteins become enzymes that speed up chemical reactions in your body. Others become structural supports, like the collagen that gives strength to your skin and bones. Still others become transporters, carrying vital cargo through your bloodstream. The process of turning RNA into protein is called translation. Your ribosomes are constantly translating genetic messages into the building blocks of life. Right now, millions of ribosomes throughout your body are reading RNA and building proteins with perfect precision. But your cellular library contains more than just protein recipes. Some parts of your DNA hold regulatory instructions. These are like the library's index system, telling the cell which books to read and when to read them. Other sections contain what scientists once called junk DNA. But we now know this isn't junk at all. These regions help control gene activity, like dimmer switches that turn genes up or down depending on your body's needs. Your genetic library also contains historical documents. Scattered throughout your DNA are remnants of ancient viral infections, like pressed flowers between the pages of an old book. These genetic fossils tell the story of your species' long battle with disease. Some of your genes come with backup copies. If one copy becomes damaged, the backup can step in to do the job. This redundancy helps protect you from genetic errors that could cause serious problems. Your cellular libraries are not static collections gathering dust. They're dynamic, responsive, constantly adapting to your body's changing needs. When you exercise, certain genes become more active. When you sleep, others take the stage. Environmental factors can influence which books your cellular libraries choose to read. The food you eat, the air you breathe, even your emotional state can affect gene expression. Your DNA provides the text, but your life experiences help determine which chapters get read most often. Scientists have learned to visit these cellular libraries and read their contents. The Human Genome Project was like creating the first complete catalog of your genetic collection. It took thousands of scientists working for over a decade to spell out all 3 billion letters of human DNA. But cataloging the books is just the beginning. Understanding what they mean and how they work together is a much bigger challenge. Scientists estimate that humans have about 20,000 genes, but these genes can produce over 100,000 different proteins through various combinations and modifications. Your cellular libraries also contain mysteries we're still trying to solve. We know that less than 2% of your DNA actually codes for proteins. What does the other 98% do? Some of it helps regulate genes, but much remains unexplored territory. Each of your cells contains the same genetic library, yet different cells read different books. A heart cell focuses on genes for pumping blood. A brain cell concentrates on genes for electrical signaling. A stomach cell emphasizes genes for making digestive enzymes. This selective reading is what allows your body to create hundreds of different cell types from a single set of genetic instructions. It's like having one enormous cookbook but choosing different recipes depending on whether you're making breakfast, lunch, or dinner. As you sink deeper into relaxation, imagine the quiet industry of your cellular libraries. Millions of RNA messengers are copying genetic instructions. Countless ribosomes are translating those messages into the proteins that keep you alive. This ancient system of information storage and retrieval has been refined over billions of years. It's more efficient than any computer, more reliable than any machine humans have ever built. Your DNA holds not just the instructions for making you, but the wisdom of countless generations who survived long enough to pass their genes forward. You carry in your cells the accumulated knowledge of evolution itself. And soon, we'll explore how this elegant system of genetic messages copies itself, ensuring that tomorrow's cells carry the same precious library that sustains your life today. Imagine the most important document you could ever possess. A document so precious that losing it would mean losing yourself entirely. Now imagine that you must make a perfect copy of this document every time you need a new one. This is exactly what happens inside your cells every time they divide. Your DNA must be copied with absolute precision, letter by letter, spiral by spiral. Not a single mistake can be allowed, because each copy will become the complete genetic library for a brand new cell. Right now, as you rest quietly and let sleep approach, millions of your cells are preparing for this incredible task. They're gathering the molecular tools needed to perform one of life's most remarkable feats of precision. The process begins when your cell receives a signal that it's time to divide. Perhaps you've scratched your skin and need new cells to heal the wound. Maybe you're growing, and your body needs more cells to support that growth. Or simply, some of your cells have grown old and need to be replaced. Your DNA normally exists as a relaxed double spiral, twisted gently like a rope ladder. But when copying begins, special enzymes approach this ladder like careful librarians preparing to duplicate an ancient manuscript. First, the enzymes must unwind the DNA spiral. They work like tiny hands, gently separating the twisted strands. As they unwind the double helix, the two strands pull apart like opening a zipper, revealing the sequence of genetic letters inside. This unwinding creates what scientists call a replication fork. Picture the shape your hair makes when you part it down the middle. The DNA forms a similar Y-shaped structure, with two separated strands branching away from the point where unwinding begins. Now the real magic happens. Enzymes called DNA polymerases move along each separated strand like patient scribes. They read each exposed letter and add its matching partner. Where they find an A, they add a T. Where they find a G, they add a C. These molecular copyists work with astonishing speed and accuracy. They can add about 50 new letters per second. That might sound fast, but remember, your DNA contains over 3 billion letters. Copying everything would take over 600 days if only one copyist worked at a time. So your cell employs thousands of copyists simultaneously. They start at multiple points along your DNA and work toward each other like teams of scribes copying different chapters of the same book. When they meet, they hand off their work seamlessly, creating one continuous, perfect copy. The precision of this copying process is almost beyond belief. DNA polymerases make mistakes less than once in every billion letters they copy. To put this in perspective, if you typed out your entire genome, you would need to fill about 1,000 books the size of a typical novel. These molecular scribes would make only 3 typing errors in all 1,000 books. But even 3 errors would be too many for something as important as your genetic code. So your cells have proofreading systems that check the work as it's being done. Special enzymes scan the newly copied DNA like careful editors, looking for any mistakes. When they find an error, they remove the incorrect letter and replace it with the right one. This proofreading system catches almost all the remaining mistakes, reducing the error rate to less than 1 in 10 billion letters. Sometimes the proofreading enzymes find more serious problems. Perhaps the DNA template was damaged, or the copying machinery got stuck. In these cases, other repair systems step in, like master craftsmen called to fix a delicate piece of clockwork. Your cells have dozens of different DNA repair systems, each specialized for fixing particular types of damage. Some repair breaks in the DNA backbone. Others fix letters that have been chemically altered. Still others remove entire sections of damaged DNA and rebuild them from scratch. As the copying continues, something beautiful begins to emerge. What started as one double helix gradually becomes 2 identical double helices. Each new helix contains one original strand and one newly copied strand, like a wise teacher paired with an eager student. This method of copying is called semi-conservative replication. Half of each new DNA molecule is conserved from the original, while half is newly made. It's an elegant solution that ensures both accuracy and continuity across generations. The copying process doesn't happen all at once throughout your entire genome. Different parts of your DNA are copied at different times, in a carefully orchestrated sequence. Some regions that contain essential genes are copied early. Others that are less critical wait their turn patiently. Your cells must also solve a unique puzzle at the very end of your DNA. These regions, called telomeres, are like the plastic tips on shoelaces. They protect your chromosomes from fraying, but they present a special challenge for the copying machinery. Normal DNA copying can't quite reach the very ends of chromosomes. Each time a cell divides, the telomeres get slightly shorter. This gradual shortening acts like a cellular clock, counting down the number of times a cell can divide. Most of your cells can divide about 50 to 70 times before their telomeres become too short. This limit helps protect you from cancer because it prevents most cells from dividing indefinitely. But it also contributes to aging, as your body gradually loses its ability to replace worn-out cells. Some special cells, like stem cells, produce an enzyme called telomerase that can rebuild telomeres. This allows these cells to divide many more times, providing your body with a renewable source of fresh cells throughout your life. As DNA copying nears completion, quality control systems perform one final check. They ensure that each chromosome has been copied exactly once, no more, no less. They verify that the new DNA is properly packaged and ready for cell division. The entire copying process typically takes several hours for human cells. During this time, your cell suspends most of its other activities and focuses entirely on this crucial task. It's like a library closing to the public while the staff works through the night to create a complete duplicate collection. When copying is finally complete, your cell contains two identical sets of genetic instructions. Soon, it will divide these sets between two daughter cells, each receiving a perfect copy of your genetic library. This process happens billions of times throughout your lifetime. Every healing cut, every growth spurt, every replacement of worn-out tissue depends on this remarkable ability to copy genetic information with near-perfect accuracy. As you drift toward sleep, your cellular copyists work silently in the darkness, preserving the genetic story that makes you who you are. But simply copying genes is only part of the story. Tomorrow, we'll discover how your cells decide which genes to read and when to read them. Imagine sitting in a vast concert hall as the orchestra prepares to play. The musicians hold identical sheet music, yet each will play only their part when the conductor signals. The violins rest silently while the flutes carry the melody. The drums wait patiently for their moment to thunder. This is exactly what happens inside every cell of your body. Each cell contains the same complete set of genetic sheet music, all 20,000 genes written in your DNA. But like skilled musicians, your cells know when to play their parts and when to remain silent. Right now, as you breathe slowly and let relaxation wash over you, this genetic symphony plays throughout your body. Different genes are awakening in your brain cells, your heart cells, your skin cells. Each type of cell performs its own unique melody while contributing to the harmony of your entire being. The process of deciding which genes to activate is called gene expression. Think of it as your cells choosing which books to read from their vast genetic library. A muscle cell might pull out the instruction manual for making contractile proteins. A nerve cell might select the guide for building electrical channels. But how does a cell know which genes to express? How does your heart cell know to focus on genes for pumping blood, while your stomach cell concentrates on genes for making digestive enzymes? The answer lies in a complex network of molecular switches and signals, like an intricate lighting system in a theater. These switches can turn genes on or off, dim them like soft candlelight, or brighten them like spotlights on a stage. Some of these switches are built into your DNA itself. Special regulatory sequences sit near each gene like faithful gatekeepers. These sequences determine who can access the gene and under what conditions. They read chemical signals in the cell and decide whether to allow gene expression or keep the gene locked away. Other switches come from proteins that float through your cell like messengers carrying important news. These regulatory proteins can bind to specific DNA sequences and either encourage or discourage gene expression. They act like conductors, signaling when each section of the genetic orchestra should begin or end their performance. Your cells also respond to signals from the outside world. When you step into bright sunlight, your skin cells receive chemical messages that turn on genes for making protective pigments. When you eat a meal, your pancreas cells detect rising blood sugar and activate genes for producing insulin. Even your emotions can influence gene expression. When you feel stressed, your brain releases hormones that travel through your bloodstream and affect gene activity in cells throughout your body. Genes involved in immune function might become less active, while genes for stress response proteins spring into action. This responsiveness allows your body to adapt to changing conditions without altering your basic genetic code. It's like having a flexible recipe that can be modified depending on what ingredients are available, while still producing recognizable results. The timing of gene expression is also crucial. During development, genes must turn on and off in precise sequences, like a choreographed dance. First, genes for basic cell structure activate. Then genes for specialized functions join the performance. Finally, genes for fine-tuning and maintenance take their positions. Some genes work in the morning shift, becoming more active when you wake up. Others prefer the night shift, increasing their expression while you sleep. These daily rhythms help synchronize your body's activities with the natural cycle of day and night. Your circadian clock genes are master timekeepers that help coordinate this daily dance. They create molecular rhythms that pulse through your cells like a gentle heartbeat, telling other genes when to wake up and when to rest. Temperature can also influence gene expression. When you have a fever, the heat activates special genes that help your immune system fight infection. When you're cold, your cells turn on genes for generating more heat and conserving energy. Some genes are like emergency responders, normally quiet but ready to spring into action when needed. If you cut your finger, genes for blood clotting and wound healing immediately activate in the affected area. Once the wound heals, these genes return to their resting state. Other genes are housekeeping genes that work constantly, like janitors keeping the cellular environment clean and organized. These genes produce proteins needed for basic cellular functions like energy production, waste removal, and structural maintenance. The pattern of gene expression gives each cell type its unique identity and capabilities. Nerve cells express genes for electrical signaling and long-distance communication. Muscle cells focus on genes for contraction and movement. Immune cells prioritize genes for recognizing and fighting foreign invaders. But gene expression isn't fixed forever. Your cells can change their gene expression patterns in response to new challenges or opportunities. This flexibility is what allows your body to heal from injuries, adapt to new environments, and even rewire your brain as you learn new skills. Scientists have discovered that identical twins, who share exactly the same DNA, can have different patterns of gene expression as they age. Their life experiences gradually influence which genes are more or less active, creating subtle differences despite their identical genetic starting point. Environmental factors can sometimes cause long-lasting changes in gene expression that persist even after the original trigger is gone. These epigenetic changes are like bookmarks placed in your genetic library, marking certain pages as more or less likely to be read in the future. Some epigenetic changes can even be passed from parents to children, allowing environmental experiences to influence gene expression across generations. It's as if your ancestors' experiences left gentle notes in the margins of your genetic textbook, subtly influencing how you read the same passages. Your cells use various mechanisms to remember which genes should be active or inactive. Some involve chemical modifications to the DNA itself, like adding small molecular tags that mark genes as on or off. Others involve changes to the proteins around which DNA is wrapped, altering how accessible different genes are to the cellular machinery. These memory systems help maintain cell identity over time. Once a cell has committed to becoming a heart cell or a brain cell, it tends to maintain that identity by keeping the appropriate genes active and the inappropriate ones silent. But the system isn't completely rigid. Under special circumstances, cells can be reprogrammed to change their identity. Scientists have learned to add specific combinations of regulatory proteins to mature cells, causing them to revert to a more flexible state where they can become. This reprogramming ability offers hope for treating diseases and injuries by converting readily available cells into the specific types needed for repair. It's like teaching a violinist to play the drums by changing which parts of the musical score they pay attention to. As you settle deeper into rest, your own genetic orchestra continues its nightly performance. Some genes that were active during your waking hours are gradually quieting down. Others that work best during sleep are beginning to stir, preparing for their nocturnal duties. Your body knows this rhythm intimately, this ancient dance between genes and time, between cellular purpose and environmental need. Soon, we'll explore how humans have learned to choreograph new movements in this dance, editing the very letters that determine which melodies your genes can play. Imagine holding a pencil that could erase any mistake in the most important book ever written. A pencil so precise it could change a single letter without disturbing the words around it. So gentle it could rewrite history without anyone noticing the edit had been made. This magical pencil exists, not in your hand, but in laboratories around the world. Scientists call it CRISPR, and it's revolutionizing our ability to edit the genetic code that shapes all life on Earth. CRISPR sounds like a kitchen gadget, and in some ways, that's exactly what it is. The name stands for something complicated, but think of it as molecular scissors combined with a GPS system. These scissors can find any specific sequence of genetic letters in your DNA and cut precisely at that location. The story of CRISPR begins not in a laboratory, but in the ancient oceans. Billions of years ago, bacteria faced a constant threat from viruses that wanted to hijack their cellular machinery. Like any living thing under attack, the bacteria evolved defenses. Some bacteria developed a remarkable immune system. When a virus infected them, they would capture pieces of the viral DNA and store them in their own genome, like keeping mugshots of criminals. They surrounded these viral mugshots with repeated DNA sequences that acted like a filing system. Scientists named this bacterial filing system CRISPR because of its repeating pattern. But the real magic happened when the bacteria encountered the same virus again. They would match the stored mugshot to the invading virus and send out molecular scissors to cut up the viral DNA. These bacterial immune systems worked with extraordinary precision. The scissors, called Cas proteins, could find their target among millions of DNA letters and cut in exactly the right place. It was like finding one specific word in a library of books and erasing just that word, leaving everything else untouched. In 2012, scientists realized they could reprogram this bacterial system to edit any DNA they wanted. They could give the molecular scissors new instructions, telling them to find and cut any genetic sequence. It was like teaching a guard dog to recognize a new intruder. The process works with elegant simplicity. Scientists create a guide RNA that matches the DNA sequence they want to edit. This guide RNA acts like a bloodhound, leading the Cas scissors through the maze of genetic letters until they find their target. When the scissors find their match, they make a clean cut through both strands of the DNA double helix. This cut triggers the cell's natural repair systems, which rush to fix the break. But here's where the real magic happens. Scientists can influence how the cell repairs the cut. They can simply let the cell stitch the break back together, which often introduces small changes that disable a problematic gene. Or they can provide a template for repair, like giving the cell a patch to sew into the fabric of its DNA. Using this template approach, scientists can insert new genetic material or correct mistakes with letter-perfect precision. They can fix the single-letter typos that cause genetic diseases. They can add entirely new capabilities to cells. They can even delete harmful sequences completely. The applications seem almost limitless. Scientists have used CRISPR to create crops that resist disease and tolerate drought. They've programmed immune cells to better fight cancer. They've corrected genetic mutations in laboratory animals, curing diseases that seemed incurable. But perhaps the most profound application is editing human embryos. In these early stages of development, CRISPR edits would be passed on to every cell in the resulting person. Such changes would become part of their genetic legacy, inherited by their children and their children's children. This possibility brings us to the edge of directing human evolution. For the first time in history, we could intentionally alter the genetic instructions that have been refined by millions of years of natural selection. We could add improvements, remove flaws, or enhance capabilities. CRISPR works so well because it hijacks the cell's own machinery. The scissors are made of the same materials as other cellular proteins. The guide RNA speaks the cell's native language. The repair systems that fix the cuts are the same ones that normally maintain genetic integrity. This compatibility means CRISPR edits are usually invisible to the cell. The edited genes function normally, express themselves naturally, and pass through cell divisions without problems. It's like seamlessly replacing a word in a sentence without changing the meaning or flow of the paragraph. The precision of CRISPR continues to improve. Early versions sometimes cut at unintended locations, like scissors that occasionally snip the wrong thread. Newer versions are more accurate, with guide systems that double-check their targets before cutting. Scientists have also developed gentler versions of CRISPR that don't cut DNA at all. These base editors can change individual letters without breaking the double helix. They work like molecular pencil erasers, rubbing out one letter and writing in another. Prime editors represent the newest evolution of this technology. They can insert, delete, or replace larger sections of DNA with even greater precision. They're like word processors for genetic text, capable of sophisticated editing operations that earlier versions couldn't perform. The speed of CRISPR development has been breathtaking. What once took months or years of genetic engineering can now be accomplished in days or weeks. The technology has democratized genetic editing, making it accessible to laboratories around the world. But with this power comes profound responsibility. Every edit to the human genome carries consequences that might not become apparent for years or decades. Changes made to embryos will affect not just the resulting person, but potentially all their descendants. The scientific community continues to debate the ethics of human genetic editing. Some argue that we have a moral obligation to use this technology to prevent genetic diseases. Others worry about unintended consequences or the creation of genetic inequalities in society. Meanwhile, the technology itself continues to evolve. Scientists are developing new versions of CRISPR that can edit genes temporarily, allowing for reversible changes. Others are working on systems that can edit multiple genes simultaneously, like changing several words in a sentence at once. CRISPR has also revealed new mysteries about genetic function. As scientists edit genes and observe the results, they're discovering that our understanding of genetic networks is still incomplete. Some genes have multiple functions that only become apparent when they're removed. Others work in complex partnerships that are disrupted by seemingly minor changes. The future may bring CRISPR-based medicines that can be taken like pills, delivering genetic edits to specific tissues without affecting the rest of the body. Researchers envision treatments that could cure genetic diseases in living patients, not just prevent them in future generations. As you rest in the quiet darkness, consider the molecular scissors working in laboratories around the world, carefully editing the genetic code that defines life itself. Each precise cut represents both incredible opportunity and weighty responsibility. The power to rewrite genetic instructions brings us face to face with questions that previous generations never had to answer. How should we shape the genetic future of our species? What wisdom do we need to guide evolution's next chapter? Deep within your genetic code lies a museum of time. Every gene carries whispers of the ancient past, memories of creatures that lived and died millions of years before you were born. As you settle into the gentle rhythm of your breathing, feel the weight of this evolutionary history flowing through your veins. Your spine remembers when your ancestors were fish swimming in prehistoric oceans. The same genes that built their backbone now build yours, following instructions refined over 400 million years. When you were developing in your mother's womb, these ancient blueprints briefly gave you structures that looked remarkably like gill slits and a tail. Evolution is life's greatest artist, working with infinite patience across vast stretches of time. It sculpts with genetic clay, trying endless variations, keeping what works and discarding what doesn't. Each successful change gets passed down through generations, slowly building complexity from simplicity. But evolution cannot see the future. It doesn't plan ahead or design with purpose. It simply responds to the pressures of each moment, favoring traits that help organisms survive and reproduce in their current environment. What we see as elegant design is actually the accumulated result of countless small improvements tested over millions of years. Your eyes demonstrate this patient artistry. The first light-sensitive cells evolved over 500 million years ago, simple patches that could barely distinguish light from dark. Gradually, these patches deepened into cups, then developed lenses, then gained the ability to focus and track movement. Today, your eyes contain over 100 million light-detecting cells, each connected to your brain through an intricate network of nerves. This incredible complexity didn't appear overnight. It emerged through countless generations of tiny improvements, each one providing a small survival advantage. The same story unfolds throughout your body. Your immune system remembers ancient battles with bacteria and viruses. Your digestive system carries adaptations from ancestors who ate raw meat, bitter plants, and foods that would poison you today. Your brain structure echoes the neural arrangements of reptiles, mammals, and early primates. Evolution works through random mutations and natural selection. Occasionally, a copying error changes a genetic letter. Most of these changes are harmful or neutral, but rarely, a mutation provides an advantage. Individuals with beneficial mutations are more likely to survive and have offspring, gradually spreading the helpful change through the population. This process seems impossibly slow when measured against human timescales. Significant evolutionary changes often require thousands or millions of years. The development of human language, complex tool use, and abstract thinking unfolded over hundreds of thousands of years of gradual refinement. But genetic engineering allows us to accomplish in years what evolution might take millennia to achieve. We can directly insert beneficial genes, remove harmful ones, or create entirely new genetic combinations that would never arise naturally. We've compressed millions of years of evolutionary time into laboratory experiments. This acceleration creates both tremendous opportunity and profound risk. Evolution's slow pace allows ecosystems to adapt gradually to new species and traits. When we rapidly introduce genetic changes, we may disrupt carefully balanced relationships that took millions of years to develop. Consider how evolution shaped your relationship with the microorganisms living in your body. Over countless generations, your ancestors developed partnerships with beneficial bacteria while learning to fight off harmful ones. This delicate balance supports your digestion, strengthens your immune system, and influences your mental health. Genetic modifications that seem purely beneficial might have unexpected effects on these ancient partnerships. A gene that makes you resistant to one disease might make you more vulnerable to another. A modification that improves one aspect of your health might compromise a different system in ways we don't yet understand. Evolution also maintains genetic diversity within populations. This diversity acts like an insurance policy, ensuring that some individuals will survive even if environmental conditions change dramatically. When we engineer specific traits into large numbers of people, we might accidentally reduce this protective diversity. Yet evolution itself is not always gentle or wise. Many genetic diseases persist because they provided advantages in certain environments. The gene for sickle cell anemia protects against malaria in some populations, but causes terrible suffering in others. Evolution preserved this gene because its benefits outweighed its costs in malarial regions. Human genetic engineering could eliminate such evolutionary compromises. We might remove genes that cause genetic diseases while preserving their protective effects through different mechanisms. We could enhance beneficial traits without accepting the harmful side effects that evolution typically requires. The question becomes whether we have enough wisdom to guide our own evolution responsibly. Evolution has tested every genetic change against the harsh realities of survival and reproduction. Our laboratory tests, no matter how sophisticated, cannot replicate the full complexity of natural selection. Some scientists argue that we should proceed cautiously, making only small changes and carefully monitoring their effects over many generations. Others believe that the potential benefits of genetic enhancement outweigh the risks, especially when used to eliminate serious diseases. The debate reflects deeper questions about human nature and our place in the natural world. Are we separate from evolution, capable of directing it through conscious choice? Or are we still subject to evolutionary forces, with genetic engineering simply representing the latest stage in our species' development? Perhaps both perspectives contain truth. We have clearly transcended many evolutionary limitations through technology, medicine, and social cooperation. We can survive in environments where our ancestors would have perished. We can treat diseases that would have eliminated entire bloodlines. But we remain biological creatures, shaped by millions of years of evolutionary history. Our emotions, instincts, and basic needs still reflect the ancient pressures that mold our ancestors. Even our ability to develop genetic engineering emerged from cognitive capabilities that evolution slowly refined over millennia. The fossil record shows that evolution sometimes accelerates during periods of environmental change or opportunity. New ecological niches open up, and life rapidly diversifies to fill them. Perhaps we're living through such a period now, with genetic engineering representing evolution's response to the challenges and opportunities of our technological age. Your genome carries the wisdom of countless generations who survived long enough to reproduce. Each gene represents a successful solution to some ancient challenge. This accumulated knowledge spans everything from fighting infections to processing complex foods to recognizing faces in dim light. As we develop the power to edit our genetic heritage, we must remember that we're not starting from scratch. We're modifying a system refined by millions of years of testing under the most demanding conditions imaginable. Every change we make should be informed by deep respect for the evolutionary wisdom encoded in our genes. The future may bring genetic modifications that help our species adapt to climate change, resist new diseases, or explore environments beyond Earth. Such changes would represent conscious evolution, guided by human intelligence rather than random mutation and natural selection. But beneath these future modifications will still flow the ancient genetic rivers that connect us to all life on Earth. Evolution's patient hand will continue to shape us, even as we learn to guide its work with tools it could never have imagined. Soon, we'll explore how genetic engineering is already transforming the microscopic partners that share your body, creating new possibilities for health and healing in the hidden world of your microbiome. You are not alone in your body. As you breathe quietly and let your muscles relax, trillions of microscopic companions share this moment with you. They live on your skin, in your mouth, throughout your digestive system. They outnumber your own cells by 10 to 1. These tiny neighbors are bacteria, and they're writing their own genetic stories inside you. Each bacterial cell carries its own DNA, its own collection of genes, its own molecular machinery for survival. Together, they form an invisible ecosystem more complex than any rainforest. Scientists call this hidden world your microbiome. It's like a secret garden that grows inside you, populated by hundreds of different species of bacteria. Some have been your family's companions for thousands of generations. Others are recent arrivals that joined your microbial community just days ago. Your microbiome began forming the moment you were born. As you passed through the birth canal, your mother's bacteria became your first microscopic inheritance. Every surface you touched, every person who held you, every breath of air you took added new species to your growing collection. Now, decades later, your bacterial garden has matured into a stable ecosystem. The bacteria in your gut help digest foods your own enzymes cannot break down. They produce vitamins your body needs but cannot make. They train your immune system to distinguish friend from foe. The bacteria on your skin form a protective barrier against harmful microorganisms. They produce natural antibiotics that kill dangerous invaders. They maintain the slightly acidic environment that keeps your skin healthy and resistant to infection. But your microbiome does more than just protect and nourish you. Recent research suggests these bacteria can influence your mood, your sleep patterns, even your food cravings. They communicate with your brain through chemical signals, participating in conversations you never knew were happening. This discovery has opened new possibilities for genetic engineering. Instead of modifying your own genes, scientists are learning to modify the genes of your bacterial companions. They're turning your microbiome into a living pharmacy, capable of producing medicines exactly where your body needs them. Imagine bacteria engineered to detect inflammation in your gut and respond by producing anti-inflammatory compounds. Or skin bacteria modified to synthesize sunscreen when they sense ultraviolet light. These microscopic factories could work around the clock, manufacturing personalized treatments from within your own body. Scientists have already created bacteria that can detect and destroy cancer cells. These engineered microorganisms patrol your bloodstream like microscopic guards, searching for the specific chemical signatures that cancer cells release. When they find their target, they produce toxins that kill only the cancer cells, leaving healthy tissue unharmed. Other researchers are developing bacteria that can break down harmful substances in your body. These living filters could neutralize environmental toxins, process excess cholesterol, or even consume the proteins that accumulate in Alzheimer's disease. The engineering process begins with understanding what your natural bacteria already do well. Scientists study the genetic networks that allow bacteria to sense their environment, process nutrients, and produce useful compounds. Then they modify these networks to create new capabilities. CRISPR, the same molecular scissors used to edit human genes, works beautifully in bacteria. Bacterial cells readily accept new genetic instructions and begin following them within hours. They can be programmed to turn specific genes on or off in response to chemical signals, light, temperature, or other environmental cues. Some engineered bacteria are designed to self-destruct after completing their mission. They carry genetic kill switches that activate after a predetermined time, ensuring they don't persist in your body longer than necessary. This built-in obsolescence addresses safety concerns about releasing modified organisms into human ecosystems. The speed of bacterial reproduction makes them ideal for genetic engineering experiments. A bacterium can divide every 20 minutes under favorable conditions, creating millions of descendants in a single day. This rapid multiplication allows scientists to test genetic modifications quickly and observe their effects across many generations. But working with your microbiome requires delicate balance. The bacterial species in your gut have spent millions of years learning to coexist. They've developed complex relationships of cooperation and competition that maintain the overall health of the ecosystem. Introducing new engineered bacteria into this established community is like adding a new species to a mature forest. The newcomers must find their ecological niche without disrupting the existing balance. They must compete for resources while avoiding harm from established residents. Scientists are learning to design engineered bacteria that integrate smoothly into existing microbial communities. Some are modified versions of species already present in your microbiome. Others are designed to fill vacant ecological niches or provide services that no natural bacteria currently offer. Your bacterial companions also serve as early warning systems for your health. Changes in your microbiome often precede the development of diseases by months or years. Scientists are developing diagnostic tools that can read these microbial signals like a medical crystal ball. Future treatments might involve regularly monitoring your microbiome and making small adjustments to maintain optimal health. Like a gardener who adds fertilizer here, removes weeds there, and plants new species to fill empty spaces, doctors could tend your bacterial garden with precision and care. The possibilities extend beyond medicine. Bacteria in your gut could be engineered to produce nutrients that are missing from your diet. Skin bacteria might synthesize cosmetic compounds that keep you looking young. Oral bacteria could prevent tooth decay by neutralizing acids before they damage your enamel. Some researchers envision bacteria that could help humans adapt to extreme environments. Microorganisms engineered to thrive in high radiation might protect astronauts during long space flights. Bacteria that process unusual nutrients could help people survive in hostile climates or resource-scarce regions. Your microbiome represents a new frontier for genetic engineering, one that exists entirely within your own body. As scientists learn to program these microscopic partners, they're discovering possibilities that extend far beyond traditional medicine. But perhaps the most remarkable aspect of this microbial world is how it challenges our understanding of individual identity. Where do you end and your bacteria begin? Are you a single organism, or a walking ecosystem? These questions become even more complex as we prepare to engineer entirely artificial life forms, creatures that exist nowhere in nature's existing catalogue. In laboratories around the world, something extraordinary is stirring. Scientists are not just editing the genetic code that nature has written over billions of years. They are writing entirely new chapters in the book of life, creating organisms that have never existed before. Picture a writer sitting before a blank page, but instead of crafting stories with words, they're composing life itself with genetic letters. Each sentence they write becomes a living cell. Each paragraph becomes a new capability. Each chapter becomes a solution to problems that have puzzled humanity for generations. This is synthetic biology, the art of designing life from scratch. It begins with the same four-letter alphabet that writes your DNA, but arranges those letters in combinations that nature never imagined. Like composers creating new melodies from familiar notes, scientists are creating new forms of life from familiar genetic building blocks. The first synthetic organism was remarkably simple. Scientists took a bacterium and gradually replaced its natural DNA with an entirely artificial genome. They wrote the genetic code on computers, synthesized it in laboratories, and breathed digital life into cellular form. This synthetic bacterium could eat, grow, and reproduce just like its natural cousins. But its genetic code contained something no natural organism had ever possessed: watermarks written by its human creators. Hidden in the DNA were coded messages, like signatures on a work of art. Since that first synthetic cell drew breath, the field has exploded with possibilities. Scientists are now designing organisms to tackle challenges that evolution never prepared life to handle. They're creating living factories that can produce medicines, fuels, and materials with precision that surpasses any industrial process. Imagine bacteria engineered to eat plastic waste and convert it into useful chemicals. These synthetic organisms could help clean up the oceans while producing valuable compounds. They would work tirelessly, reproducing themselves and expanding their cleanup operations without human intervention. Other synthetic organisms are being designed to capture carbon dioxide from the atmosphere and transform it into useful products. These living carbon sinks could help slow climate change while producing the raw materials for plastics, fuels, and other essential chemicals. The process of creating synthetic life begins with understanding what you want the organism to do. Scientists first identify the specific functions they need, then search through nature's genetic library for biological parts that could serve those functions. These biological parts work like components in an electronic circuit. Some parts act as sensors, detecting specific chemicals or environmental conditions. Others function as switches, turning cellular processes on or off. Still others serve as motors, pumps, or manufacturing systems. Scientists collect these functional parts from different organisms and combine them in new ways. They might take a light sensor from a plant, a chemical detector from a bacterium, and a protein manufacturing system from a yeast cell. When properly connected, these parts work together to create entirely new capabilities. The design process requires careful attention to how biological parts interact with each other. Like engineers designing a complex machine, synthetic biologists must ensure that all components work together harmoniously. A sensor that's too sensitive might overwhelm the system. A switch that turns on too slowly might miss important signals. Computer models help scientists predict how their designs will behave before building them. These simulations can test thousands of different configurations, identifying the most promising combinations and revealing potential problems before they occur. Once a design is complete, scientists must translate it into actual DNA. They write genetic code that specifies each biological part and how they should be connected. Modern DNA synthesis technology can then manufacture these genetic instructions with remarkable precision. The synthetic DNA is introduced into living cells, usually bacteria or yeast that serve as cellular chassis. These host cells provide the basic machinery needed for life, while the synthetic DNA programs them with new capabilities. Testing synthetic organisms requires patience and careful observation. Scientists must verify that their creations behave as expected and don't produce harmful side effects. They monitor growth rates, chemical production, and cellular health across many generations. Sometimes synthetic organisms exceed their designers' expectations. The interplay between engineered parts and natural cellular machinery can produce unexpected behaviors. These surprises often lead to new insights about how life works and suggest improvements for future designs. Safety is paramount in synthetic biology. Scientists build multiple safeguards into their creations to prevent them from surviving outside controlled laboratory conditions. These biological kill switches ensure that synthetic organisms cannot establish themselves in natural ecosystems. Some synthetic organisms are designed to require artificial nutrients that don't exist in nature. Others carry genetic circuits that cause them to self-destruct if they lose contact with their human controllers. These precautions help contain synthetic life within the boundaries where it's intended to work. The applications of synthetic biology continue to expand. Scientists have created organisms that produce life-saving medicines, including insulin for diabetes and antimalarial drugs. Others manufacture industrial chemicals, reducing our dependence on petroleum-based processes. Synthetic biology is also revolutionizing agriculture. Scientists are designing plants with enhanced nutritional content, improved disease resistance, and the ability to grow in harsh climates. These crops could help feed a growing global population while reducing environmental impact. The field extends beyond Earth as well. Scientists are developing synthetic organisms that could help establish human settlements on Mars. These organisms might produce oxygen from the Martian atmosphere, manufacture building materials from local resources, or process waste into useful compounds. Future synthetic organisms might be designed to interface directly with human biology. Therapeutic bacteria could be programmed to detect disease markers in your body and respond by producing targeted treatments. These living medicines would provide personalized healthcare from within your own cells. The boundary between natural and artificial life is beginning to blur. Synthetic organisms incorporate natural biological parts but arrange them in unnatural combinations. They follow the same fundamental principles as all life but serve purposes that evolution never selected for. This fusion of natural and artificial raises profound questions about the nature of life itself. What makes something alive? Can we consider synthetic organisms truly living, or are they elaborate biological machines? Do the intentions of their creators change their essential nature? These questions become even more complex when we consider the possibility of creating synthetic versions of human cells or tissues. The techniques that work in bacteria and simple organisms could someday be applied to engineer human biology itself. The quiet hum of laboratory equipment masks a revolution in progress. Each synthetic organism represents a small step toward a future where life itself becomes programmable, where biological systems can be designed to solve humanity's greatest challenges. As these artificial creatures multiply in their carefully controlled environments, they pose questions that extend far beyond science into the realm of ethics, responsibility, and the very meaning of creation. In the quiet moments before sleep, when your mind settles and your thoughts turn inward, consider the weight of the choices we now face as a species. For the first time in human history, we hold the power to rewrite the genetic instructions that have guided life for billions of years. This power brings with it questions that keep philosophers awake at night and ethicists pacing in their studies. Every edit we make to the human genome will echo through generations yet unborn. Every synthetic organism we create will interact with ecosystems in ways we cannot fully predict. The children born tomorrow might carry genetic modifications that protect them from diseases that have plagued humanity for millennia. Their children, in turn, might inherit these changes along with others we cannot yet imagine. Each edit becomes part of humanity's permanent genetic legacy. Consider the parent who discovers their unborn child carries genes for a devastating disease. But even this seemingly clear-cut case raises difficult questions. Which genetic variations should we consider diseases worthy of correction? Where do we draw the line between treating illness and enhancing normal human capabilities? Who gets to decide what constitutes a genetic flaw? Some genetic conditions bring both advantages and disadvantages. The gene variants that cause sickle cell disease also protect against malaria. The genetic differences that contribute to autism may also enhance certain cognitive abilities. When we edit these genes, we might eliminate both the problems and the unexpected benefits. Cultural perspectives further complicate these decisions. The deaf community has argued that deafness is not a disability to be cured, but a cultural identity to be preserved. Some people with dwarfism embrace their condition as part of who they are. Genetic editing that eliminates these conditions might also eliminate rich traditions and communities built around them. The question of enhancement versus treatment becomes even more complex when we consider genetic modifications that don't address medical conditions at all. Should parents be allowed to edit their children's genes to make them taller, stronger, or more intelligent? Should society permit genetic modifications that could give some people significant advantages over others? These enhancements might create new forms of inequality more profound than any we've seen before. Genetic advantages, once acquired, would be permanent and heritable. Families wealthy enough to afford genetic enhancements might pass those advantages to their children and grandchildren, creating biological class divisions that persist across generations. Yet refusing to allow genetic enhancements might also be ethically problematic. If we can prevent genetic diseases, shouldn't we also be able to enhance genetic strengths? If we can correct genetic flaws, why not improve upon genetic gifts? The line between treatment and enhancement often blurs when examined closely. The global nature of genetic research adds another layer of complexity. Scientific advances in one country affect people everywhere, but different nations have different ethical standards and regulatory approaches. A genetic modification banned in one country might be developed and offered in another. This patchwork of regulations could create genetic tourism, where wealthy individuals travel to countries with looser restrictions to obtain genetic modifications for themselves or their children. Such practices could undermine efforts to ensure genetic editing is used safely and equitably. Religious and philosophical traditions offer diverse perspectives on genetic modification. Some view the human genome as sacred, created by divine will and not meant to be altered by human hands. Others see genetic editing as a moral obligation, a way to use God-given intelligence to reduce suffering and improve life. Secular ethical frameworks also disagree about genetic modification. Some emphasize individual freedom and the right of parents to make decisions about their children's genetic future. Others focus on social justice and the need to prevent genetic technologies from increasing inequality. The consent of future generations presents perhaps the most challenging ethical puzzle. When we edit the genes of embryos, we make irreversible decisions for people who cannot speak for themselves. These future individuals will live their entire lives with genetic modifications they never chose. Yet all parental decisions shape children's futures in permanent ways. Parents choose where their children live, what education they receive, what values they learn. These choices profoundly influence who their children become, often more than genetic modifications would. Is editing genes fundamentally different from other parental decisions? The pace of scientific progress makes ethical deliberation especially urgent. Researchers are developing new genetic editing techniques faster than ethicists can evaluate their implications. The technology to edit human embryos already exists, and the techniques continue to improve rapidly. Some scientists argue that we should proceed cautiously with genetic editing, taking time to fully understand the risks and develop appropriate safeguards. Others worry that excessive caution will delay treatments that could save lives and reduce suffering for millions of people. The international scientific community has attempted to establish guidelines for genetic research, but these voluntary standards have limited enforcement power. Some researchers have already violated these guidelines, editing human embryos in ways that many colleagues consider premature and irresponsible. Public opinion on genetic editing varies widely and continues to evolve as people learn more about the technology. Surveys suggest that most people support genetic modifications to treat serious diseases, but opinion is more divided on genetic enhancements. Trust in scientists and institutions also influences how people view genetic editing. Democratic societies face the challenge of making collective decisions about genetic technologies while respecting diverse viewpoints and values. These decisions will shape not only our species' genetic future but also our relationship with nature and our understanding of human identity. The environmental implications of genetic modification extend beyond human applications. Genetically modified crops and synthetic organisms could have far-reaching effects on ecosystems. Once released into the environment, these organisms might interact with natural species in unpredictable ways. Gene drives, which can spread genetic modifications rapidly through wild populations, present particularly complex ethical challenges. These tools could eliminate disease-carrying mosquitoes or invasive species, but they could also irreversibly alter entire ecosystems. The effects would likely cross national borders, making these technologies a global concern. The reversibility of genetic modifications influences their ethical acceptability. Some genetic changes can be undone if problems arise, while others become permanent parts of the human genome. The permanence of germline editing, which affects sperm and egg cells, makes these modifications especially ethically significant. Economic considerations also shape the ethics of genetic modification. The development of genetic therapies requires enormous investments in research and development. Companies need to recover these costs, which often makes new treatments expensive. The high cost of genetic therapies raises questions about access and fairness. Insurance companies might eventually require genetic testing or offer different rates based on genetic profiles. Employers might prefer to hire individuals with certain genetic advantages. These practices could create new forms of discrimination based on genetic characteristics. As you contemplate these weighty questions in the growing quiet of night, remember that every generation faces ethical challenges that seem overwhelming. Our ancestors grappled with the morality of industrialization, nuclear weapons, and environmental destruction. Now we face the ethics of genetic modification. The responsibility we bear is profound, but so is our capacity for wisdom and moral growth. The very ability to ask these questions thoughtfully is itself a form of progress. The answers we choose will determine not just the genetic future of our species, but the kind of moral beings we decide to become in the process. Close your eyes now and feel the gentle rhythm of your breathing. With each breath, you participate in an ancient dance that began billions of years ago and will continue long after you have become part of memory. Your chest rises and falls with the same pattern that sustained your ancestors and will sustain generations yet to come. In the growing darkness behind your eyelids, your genes are not resting. They work through the night like devoted gardeners, tending the molecular landscape of your cells. They repair the small damages that accumulated during your day. They prepare the cellular machinery for tomorrow's needs. They whisper instructions that keep your heart beating, your lungs breathing, your dreams flowing. Your genetic legacy is not simply the DNA you might pass to your children. It is the sum of all the moments when your genes expressed themselves in service of life. Every protein they directed your cells to build. Every immune response they orchestrated. Every healing process they guided. This is your genetic autobiography, written in molecular ink across the pages of time. Tonight, as sleep approaches, your genes are writing new chapters in this autobiography. In your brain, genes for memory consolidation are activating, helping transform today's experiences into lasting memories. Growth hormone genes in your pituitary gland are preparing to release their chemical signals, promoting healing and renewal throughout your body. Your skin cells are following genetic programs for overnight repair, fixing damage from ultraviolet light and environmental toxins. Your liver cells are expressing genes for detoxification, filtering your blood and preparing your body for tomorrow's challenges. Your muscle cells are reading genetic instructions for protein synthesis, rebuilding, and strengthening the fibers that moved you through today. This nightly genetic symphony has played for millions of years in countless species. The same basic melodies that orchestrate your sleep also guided the rest of your ancestors, from the earliest mammals to the humans who first looked up at stars and wondered about their place in the cosmic story. But you live in a unique moment in this ancient tale. You are among the first humans to understand the genetic language that writes itself through your cells. You are part of the first generation that can read, edit, and even rewrite the genetic instructions that have shaped life since its beginning. Your children, or their children, might carry genetic modifications that protect them from diseases that have troubled humanity for millennia. They might possess enhanced abilities to resist environmental toxins, process different nutrients, or adapt to changing climates. They might even carry genetic tools that help them survive in environments beyond Earth. These possibilities rest not just in future laboratories, but in the decisions your generation makes about how to use the genetic knowledge you have gained. The ethical frameworks you establish, the safety protocols you develop, the values you choose to embed in genetic technologies will shape how future generations write their own genetic stories. Your DNA carries messages from the deep past. Embedded in your genetic code are sequences that remember when your ancestors lived in trees, when they first walked upright, when they learned to control fire and speak in complex languages. These genetic memories connect you to every human who has ever lived and to the long lineage of species that led to humanity's emergence. But your genes also dream of the future. They carry the potential for adaptations that haven't yet been needed, capabilities that might only emerge under conditions that don't currently exist. Your genetic code is not a fixed blueprint but a living document that continues to accumulate new possibilities with each generation. The bacteria in your microbiome are also dreaming genetic dreams tonight. They exchange genetic material among themselves, sharing survival strategies and passing beneficial mutations through their communities. Some of these microbial genetic innovations might eventually become integrated into human genetic therapies, as scientists learn to harness the evolutionary wisdom of your smallest companions. In laboratories around the world, synthetic organisms created by human ingenuity are experiencing their own form of genetic dreams. These artificial life forms represent humanity's first attempts to write entirely new chapters in the book of life, using the same four-letter alphabet that writes your own genetic story. The genetic modifications that might one day become commonplace are still being tested in these laboratories. Scientists are learning to edit genes with increasing precision, to predict the effects of genetic changes with greater accuracy, to balance the benefits and risks of genetic interventions with growing wisdom. Your generation stands at the threshold between two eras of human existence. Behind you stretches the long history of evolution by natural selection, where genetic changes occurred randomly and were tested by the harsh realities of survival and reproduction. Ahead lies an era of conscious evolution, where genetic changes are deliberately designed to serve human purposes and values. This transition brings both tremendous opportunity and profound responsibility. The genetic tools you are developing could eliminate inherited diseases, enhance human capabilities, and help your species adapt to challenges that natural evolution could never prepare for quickly enough. Yet these same tools could also create new forms of inequality, disrupt ecological relationships that took millions of years to develop, or produce unintended consequences that only become apparent across generations. The wisdom to use genetic power responsibly is still evolving, much more slowly than the technology itself. As you sink deeper into relaxation, imagine the genetic conversations happening within your cells. Regulatory proteins are binding to specific DNA sequences, turning genes on and off in response to the changing chemistry of sleep. Repair enzymes are scanning your genetic code for damage, making corrections with molecular precision. Your telomeres, the protective caps on your chromosomes, are shortening ever so slightly with each cell division, marking the passage of cellular time. But other genetic systems are working to maintain your health and vitality, balancing the inevitable processes of aging with ongoing renewal and repair. The dreams you will soon experience might themselves be influenced by genetic variations that affect how your brain processes information during sleep. Your genes help determine how vividly you dream, how well you remember your dreams, and how sleep helps consolidate the memories that shape who you are. In your dreams tonight, you might find yourself flying, exploring impossible landscapes, or meeting people from different times and places. These dream experiences are made possible by genetic programs that temporarily suspend your brain's normal reality-checking systems, allowing imagination to flow freely through neural networks. Your genetic heritage includes the capacity for this kind of creative dreaming. The same genetic variations that make human consciousness possible also enable the flights of imagination that have led to art, music, literature, and scientific discovery. Your genes don't just build your body, they create the foundation for all human culture and creativity. Future genetic modifications might enhance human dreaming, memory, or creativity in ways that are difficult to imagine now. But they will build upon the genetic foundation that already makes you capable of wonder, curiosity, and the kind of abstract thinking that allows you to contemplate your own genetic nature. Your genetic legacy extends beyond your individual life in ways both profound and subtle. Every environmental exposure influences how your genes interact with the world around you. Every relationship you form has the potential to shape the genetic future of humanity. The children you might have will inherit not just your DNA, but also the epigenetic marks that reflect your life experiences. The stresses you've endured, the foods you've eaten, the environments you've lived in all leave molecular signatures that can influence gene expression in your offspring. This intergenerational genetic communication means that your life story becomes part of your genetic legacy in ways that previous generations never understood. Your experiences become encoded in molecular memory systems that can influence your children's development. As sleep draws near, feel yourself becoming part of this vast genetic river that flows from the ancient past toward an unknown but hopeful future. Your genes carry the accumulated wisdom of billions of years of evolution, tested and refined by countless generations of survival and adaptation. Tomorrow, when you wake, your genes will continue their work of maintaining and renewing your body. They will respond to the new day's challenges with strategies refined over millions of years of evolutionary time. They will adapt to changing conditions with the flexibility that has allowed life to persist through ice ages, asteroid impacts, and countless other challenges. Your genetic story is still being written, one day at a time, one cell division at a time, one protein synthesis at a time. You are both the author and the main character in this story, carrying forward the genetic wisdom of the past while helping to create the genetic possibilities of the future. Rest now in the knowledge that you are part of something larger than yourself, yet irreplaceably unique. Your genetic signature has never existed before and will never exist again in exactly the same form. You are a singular expression of life's endless creativity, a unique verse in the genetic poetry that has been composing itself for billions of years. In your dreams tonight, may you sense the gentle presence of all the lives that came before you and all the possibilities that stretch ahead. You are the bridge between evolution's patient work and humanity's conscious choices about the genetic future we will create together.